The project I will be presenting today is one of the newest projects in our organization, as Lisa just mentioned moments ago. This is a work in progress, so I will be giving you an update on how we are approaching it and how what's the progress right now, and also the next phases. The project is to test the combination of Senolytics and mesenchymal stem cells transplantation and determine if they have any synergistic effect in enhancing health span in mice. So for us, this is also the opportunity to build the capabilities of in vivo testing in our research center. This will allow us to test other aging interventions that we are developing here, and also to help other scientists in the longevity field with the research and testing their new interventions using our animal models. So to start, I will give you a brief background and also the rationale for our combination. So as you all know, the hallmark of aging, at least one of the hallmarks, is the accumulation of senescent cells in the body with age. And senescent cells are, of course, considered harmful because they secrete pro-inflammatory and pro-fibrotic factors that disrupts the homeostasis of normal tissues. And there is increasing studies right now that implicate senescent cells in progression and or the onset of aging-associated diseases. So this makes senescent cells an attractive target for the development of anti-aging therapeutics. So in order to give you an overview on potential approach to reduce the harmful effect of senescent cells, I would like to look at it from the developmental point of view. Most tissues have senescent cell pool from which progenitor cells and also tissue functional cells are generated, and with time, either due to the differentiation of these cells or the accumulation of damage, these cells can undergo different states all the way to senescence and also to cell death. And one of the particularity of senescent cells is that they are resistant to stress and they secrete a SASP that affect neighboring cells in the same tissue or other cells in different tissues. And because of that, they affect the function and they interfere with well-functioning of tissues. So one of the obvious approach to overcome this is by using Senolytics, and these can be either small molecules or biologics. Another approach is to use molecule that either inhibit the expression or the effect of SASP on neighboring cells. A third approach is by basically try to inhibit senescent cells formation or generation by using either molecule that inhibit damage or like caloric restrictions. There are also, as you know now, exciting opportunity for reprogramming, partial reprogramming at least, and so this is a new and promising, of course, approach, but it needs optimization to minimize the risk of cancer. Also, there is the reprogramming of senescent cells. It can be challenging actually, and because themselves are a barrier to reprogramming. But it will be interesting to see how this will unfold in the future. This brings us also to now to mesenchymal stem cell transplantation. This has been done for decades now, and it has shown that it has a beneficial effect on many aging-associated diseases. However, the mechanism of action is not well defined. Initially, it was believed that that mesenchymal stem cells transplantation lead basically the cells differentiate directly into and replace or replenish tissue functional cells. But the evidence for this was scarce, and this hypothesis is now less pursued. So the common thinking is that mesenchymal stem cells actually work in a paracrine manner by secreting factors or EVs that can actually affect the neighboring cells. And one example is, for instance, the secrete factors that induce the proliferation and differentiation of progenitor cells into functional cells. Other effects are, for instance, the induction of angiogenesis or immunomodulation, and I will also add to that that they may have possibly a xenomorphic effect that we are actually trying to investigate right now to demonstrate whether it's possible. So because mesenchymal stem cells have demonstrated a beneficial effect in various aging-associated diseases, and because they have been shown to have favorable toxicity profile, or safety profile, I mean, they constitute a good starting point for us to do combination with Senolytics. So before we start the intervention, there are a number of factors to be considered. First, one of them is the animal model to be used. Secondly, the sequence of treatment, whether we use Senolytics first or stem cells first. And the third one is what type of Senolytic to be used. And of course, the source of quality of an optimization status of mesenchymal stem cells. And the most important one actually is the biomarkers. Is how are we going to know whether the interventions are working? So we want to onboard and validate all these biomarkers in vitro and in vivo before we start the interventions. So in terms of the animal model, we will be using a CD57 black 6 mice that are naturally aged. But for the other factors, for instance, the treatment of sequence of treatment, we will use Senolytic first, then followed by mesenchymal stem cells. And the reason for that is we don't want to have senescent cells when we inject stem cells because they may affect their regenerative effects, and we don't want to have stem cells there when we inject Senolytics because Senolytics may have some side effect on stem cells. So that's why we start with Senolytic first, to eliminate senescent cells, and then we transplant mesenchymal stem cells. With regard to the type of Senolytics, of course, there have been the last few years the discovery of many Senolytics, but however, most of them are not very selective or only cell type specific. The one that stands out is Navitoclax, is the most established Senolytic so far, and it has been shown to reduce the burden of senescent cells in vivo and in vitro. However, the problem with this drug is that it has toxic effects, especially toward neutrophil, because they express the targets of this drug. So because of that, the translation of Navitoclax to the clinic can be challenging. And so in order to go around that, our collaborator, he sensitized this molecule called Navitogal or Navitoclax linked to galactose. So this makes Navitoclax inactive, and it can become active only when it go into senescent cells that express the senescence-associated beta-galactosidase. So in that case, when it's cleaved, good lysosome is cleaved, it binds to the receptor BCLXL. However, in non-senescent cells, because you don't have this enzyme, even though it gets into the cells, it will not kill them because it needs to be activated. And this is the case, for instance, in neutrophil. So it will be safe for neutrophil this way. And actually, the group that discovered this molecule, they tested neutrophil and they found it that is less toxic to these cells. This molecule has been tested in cancer and it was effective, and we will test it in aging in our model. So we tested in vitro and compared it to Navitoclax, and it does in fact kill senescent cells selectively, although a little bit less than Navitoclax itself, and that is expected because not only this molecule has to cross the membrane, but also has to go through cleavage by Navitogal. However, Navitoclax itself, it doesn't have to go through the cleavage because it's already ready to kill senescent cells. So because of this, we think at least the selectivity towards senescent cells will help in making less toxic movement for our studies. With regard to the commercially available bone marrow mesenchymal stem cells are used, and work is ongoing right now to cultivate these cells, characterize them, and make stocks of them. And also, we are also isolating EVs at the same time. Do work in optimizing these stem cells, and mostly we try to increase their survival in the host, because one of the problem that these cells have is that they have short life span in the mice. And this is has been done a simple experiment that this mesenchymal stem cell labeled, and when they're injecting the mice, they don't go more than 7 days, then they die. And one of the mechanisms by which they die is the cell death called anovakis, which is a cell death that occur in cells that are oncourage dependent. So we will be targeting this mechanism of cell death and other survival pathways to increase the survival of mesenchymal stem cells, and hopefully will increase their ability to regenerate tissue. And as I said earlier, one of the most important aspects of the study is to establish biomarkers, so we will be able to know how our interventions are working. So we are testing a variety of markers and different ways, whether with colorimetric or fluorescent assays, or immunohistochemistry, or ELISA, or qPCR, and we're testing these markers either or in tissue specimen from young and old mice to see. The goal is to really find out how specific they are, how selective, how sensitive, and how reliable. And if we can get even few markers that works very well, then we can move to really test the interventions. Doing imaging, we just acquired an imaging system that allow us to detect senescent cells either by using fluorescent probes such as CY-GAL or galactose linked to Nile blue. We will also use this machine to track mesenchymal stem cells that are transplanted, and also to detect senescent cells using this mouse model of P16 3MR that also can be linked to luciferase and detect luciferase stem cells. So just to give you an example of the staining that we're doing, this is a senescence-associated beta-galactosidase, and also we are doing EDU staining to verify that these methods are working in our hand, and the senescent cells, of course, they don't stain. This is a marker for proliferation, and also a gamma H2X, which is a marker for DNA damage. and the senescent cells stand more than non-senescent cells. And also we're doing P21 cell cycle inhibitor, and it shows same thing. It shows increase in senescent cells. Just to show you a little bit how this gamma H2X works, it actually detects DNA damage, so it should punctate into a nucleus of senescent cells, and this is what we see here. So just to zoom from the previous slide. We have also stained with the galactose linked to CY7. This is a probe that should be cleaved only in senescent cells, and CY7 can be detected. The staining is weak, it's specific, but it's weak. So we will be trying this also in vivo, but it says the weakness of this probe may be a problem going forward when we move it in vivo. We have also tested galactose linked to a Nile blue, and this is more strong, but it's less specific. So we will also try it later on. We now this is in specimen from young and old mice, 70 weeks and 12 weeks. We stain with the senescence associated beta galactocytase, and as you can see, it shows a specific staining in old mice and in area, and this is the lung, in area that is around the alveoli. The CY7 gall was not that strong. So again, it may be a problem when we do the imaging with it. We have also stained with ferritin. This is a staining in the kidney, and it was increased in old mice as opposed to young mice. And this is very encouraging. And this type of staining was based on our finding previously that the iron is increased in senescent cells and increases in tissues in general. So this is a proof that actually it's working. We have also run a SASP, senescence associated profile, secretory profile in mouse liver, compared between young and old mice. And we found, this is in this panel that we have tested here as an example, we found some factors as interleukins were not affected in a significant way. However, the P16 was very significant, and also it was consistently increased in old mice as compared to young mice. Interestingly, we found that serpin is the opposite, was decreased in old mice as compared to young mice. And so at least these two, we will use them for as in future step as a marker for intervention effects. Metal proteases, the MMP3 was increased significantly only in one mice out of 3. MMP9 was increased in 2 out of 3. So maybe when we increase the group size, hopefully we see more significance, and probably we can use them also for interventions. We have acquired this imaging machine just a few weeks ago, and we are now optimizing the probes to detect senescent cells, fluorescent probes, and this is ongoing work right now. And also we are testing it for a mouse model of P16 luciferase, and it shows it's working. But this one we can use it later if the fluorescent doesn't work, because we would like to use a non-transgenic mouse model first. So yeah, and the next steps will be to test Senolytics and stem cells, of course, but this is for Senolytic alone. The procedure will be a standard one that has been described before for Navitoclax, and basically 2 weeks treatment, 5 days on and 2 days off, and the mice will be imaged throughout the study, and at the end they will be sacrificed, and the specimen will be harvested. We will be testing all these markers that we was talking to you about, and so the primary outcome is to obtain a significant reduction in senescent cell markers and SASP. However, this if that doesn't work, this is we have to start again and try another senolytic before we go to the combination, because this is for us, this is a limiting step. So we need to find the senolytic that work, and then we can go to the next step with the combination. Otherwise, we will have to use the transgenic mouse model with three mice, because those have been already validated, and the senescence burden can be reduced in these mice. With regard to stem cell transplantation alone, the procedure again has been described, so we will just follow the same thing. We will inject the mice with a single injection, and then a month later we will analyze the markers of. Mostly we will be interested in measuring markers of pro-inflammatory and pro-fibrotic markers, but we may also do senescent cell burden markers, although we are not expecting that mesenchymal stem cells will kill senescent cells. In case this procedure, this mesenchymal stem cell do not work, we may use AVs or optimized mesenchymal stem cells that we are currently generating. With regard to the combination of senolytic and stem cells, we will do the same thing. We will do either agent alone or the combination of both. And in this time, we will, of course, we will do the imaging, of course, but we also will do physical and behavioral studies, and also we'll collect survival data. And at the end of the experiment, we will do analysis of biomarkers of senescence. And so the outcome will be to see if single agent or treatment combination will reduce the burden of senescent cells and SASP, will have any effect on physical and behavioral activities, and whether they enhance the health span or lifespan. So I hope I will have this data ready for next meeting, and I will be able to share it with you. So in conclusion, we hope this finding will lead to uncover a new approach for treating aging, or at least enhance our understanding on how this intervention work together, and how can we improve them and optimize them to increase health span. And again, this is a project that will allow us to establish the capabilities and standardize the process of in vivo preclinical testing at our research center here at the Science Research Foundation. By standardizing this process, we hope to explore additional combinations that we are developing here on site, or help other scientists with their discoveries and by using our in vivo models and platform. So with this, I would like to thank the SRF donors and project sponsors, the board and the executive team, and also the research colleagues and all the SRF staff. And I would like to thank my team member for their hard work and dedication, and also our collaborators for giving us the product that we are using to test in this study. And thank you all for attending. Thank you.